So the first question on your exam reads, you have recently started working in a research lab and have been tasked with solving a little bit of a problem that has other lab members stumped. I'm going to go ahead and be underlining and highlighting as I do this. Okay, so we've got a problem. You have an infinite supply of a polypeptide that is approximately 400 amino acids long. I'm losing my stylus, and now I got it back. 400 amino acids long. The average amino acid is about 110 Daltons. Okay, so this is our first piece of information. 400 times 0.110. And I just converted this to kilodaltons. You don't have to do that, but it might not be problematic or might not be harmful to you. Okay. Now, after that, we've got after cutting this polypeptide with the protease trypsin, okay, trypsin, which cuts after positively charged residues like arginine and lysine, you observe that when you run the reaction mixture on your SDS page, you have a total of 5 bands. Okay, so SDS page, this is another piece of information. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS page, that is a denaturing, or it's in a denaturing conditions. So what do you see whenever you're in denaturing conditions? One thing that you're going to see is you're going to basically take the 3D structure of a protein and convert it to, well, a straight chain, a 1D. And that's going to also give you a bunch of negative charges, uniform negative charge. Those are all my negative charges across the length of the polypeptide chain. And in denaturing conditions, one of the things that you're going to do is you're going to break disulfide bonds. Okay, so these disulfide bonds exist between two cysteines. But other than that, that's basically your covalent interaction that you're concerned with. You're taking this one polypeptide and you're breaking it up. Okay, so now back to the question. The bands present are approximately 23 kilodaltons, 16, 12, 10, and 6. Okay, so if I were to look at this on a SDS page, denaturing gel, it would look a little bit like this. We've got our 23, our 16, our 12, our 10, and our 6. Okay. For comparison, you collect another portion of the protein and treat that with trypsin and run it on a native page. Okay. Now trypsin is still going to cut those specific bonds. I'm going to go ahead and say SDS. But what I've got here is in non-denaturing conditions, well, as is the case with a native page, I'm not going to break those disulfide bonds. Likewise, my three-dimensional protein, well, it's going to exist as a three-dimensional protein. But we got to keep in mind, we've cut this, okay? So you collect another portion of the protein, cut it with trypsin, native page, in which non-covalent interactions are disrupted. Doesn't say anything about covalent. And notice the bands are 23, 16, and 12. Okay. What is the explanation for this discrepancy? Okay, so first of all, we have to point out what our discrepancy is. This band and this band, they're both absent. So the 10 kilodalton and the 16 kilodalton bands are both absent. Now, what's the significance of that? Well, a 10 kilodalton and a 16 kilodalton band are gone, but let's think about this. What is not happening? So basically, to answer this first question, we're looking at SDS versus native. And the difference between them is the covalent interactions are being disrupted in SDS and not being disrupted in a native page. So effectively, the answer to this question is you have a polypeptide, and I'm just going to draw this as a linear structure. We have a disulfide bond that connects two cysteine residues. And the way that trypsin's going to cut is it's going to cut here, here, here. That would be, we'd need a total of 1, 2, 3, 4. Yeah, so it would cut something like this. Now, one way that you could view this is 10 kilodaltons, this is 6, and I'm not drawing this to scale well. This is 12, and this is 16. Okay. Now, with that said, what did not change between our SDS and our native was this 23 kilodalton protein. Now, before we proceed and really analyze that, if we have something that is 400 amino acids long, each amino acid is 0.110 kilodaltons, we're talking about something, and I'm going to get my calculator out just so I don't screw up my math. 400 times 0.110, that's something that is 44 kilodaltons. Okay, so yeah, so that 44 kilodaltons, well, if this one is 16, well, we're talking about 16 plus 12 is going to be 28, plus 10 plus 6, that's going to equal 44 kilodaltons. Okay, now this 23 kilodalton protein, where the heck is that coming from? Well, let's think about our mixture. Our mixture that we, or our experiment that we basically ran was trypsin plus our polypeptide. So this 23 kilodalton band is trypsin. Now that means the rest of these are our 44 kilodalton polypeptide. So what that means is that this 16 kilodalton band, well, we got a 16 kilodalton fragment, but then we have two bands, 10 and 6, that also migrate that are 16 kilodaltons as well. So this 16 kilodalton band, if you were to see anything, any noticeable difference, it might be a little bit fatter, but it's going to be the exact same mass, or it's going to be the exact same level. Okay, so that basically answers part 1. Now part 2 of this question is based on what's presented in this problem and the tools that we have discussed in class, what would be the simplest strategy to determine the sequence of the polypeptides? Okay, so essentially what this is asking is we've got these 4 different polypeptides. How could we determine which one is the first, second, third, fourth? Okay, so one way that we could do it, and I kind of gave a little bit of latitude. So actually, let me take it a step back. For this first question, what I was looking for was something that alluded to those disulfide bonds and the fact that trypsin's not going to cut those disulfide bonds, and so a 10 kilodalton and a 6 kilodalton protein are going, or polypeptide are going to run at the same level as a 16 kilodalton band. Okay. Now, with that said, the second item here, the second part of this question was, and is best answered by, we could have used something like endopeptidase or carboxypeptidase. These are reactants that will, that will cut off the C-terminal amino acid. And so you could do that, and then since you know that this is going to cut after, or trypsin's going to cut after positively charged residues, you can basically say, well, I know that there will be one polypeptide that does not have a positively charged residue, and that's going to be my C-terminal polypeptide. And you could go through and repeat this same process. So that's one piece of evidence that will identify which is your C-terminal peptide. The other 3 polypeptides, what you'd want to do is you'd want to take the original polypeptide, since you have an infinite supply of it, and cut it with another protease like chymotrypsin or something like that and see the banding pattern, then use the carboxypeptidase. So you'd be assembling these little pieces, look at the molecular weights, and then see, okay, how does this line up with this? Cut with trypsin and chymotrypsin, or cut with trypsin, cut with chymotrypsin, and then cut both of them, or cut with both chymotrypsin and trypsin. So what I was looking for there was something alluding to using a different different protease, and or something that alluded to possibly using gel filtration chromatography to separate based on molecular weight, and then subsequently use ion exchange chromatography, because you know that there's going to be one positive charge at the end of each of these, so you could use that ion exchange chromatography to help place those individual polypeptides. All right, so that's all what I was looking for question number 1. I went ahead and I looked at each of these as equal parts, so I graded them on a scale of 0.5. So that subsequently I scaled up so that this was a test worth 100 points. If you got 0.5 or something like that, that's either indicative that you got half credit on each part, or full credit on only one part. All right, well, I'm going to be proceeding in just a moment with part 2, or question number 2.